Today's lecture, that is a continuation of this neuromuscular junctions disorder. Okay, here the continuation of these 2 years, neuromuscular junctions disorder. Here in this neuromuscular junctions disorder, last lecture we already studied about the how the neuromuscular junction is there, and how the neuromuscular junction, it means the presynaptic nerve, then the synaptic cleft and the postsynaptic nerve, it means the muscles, how those muscle get the signals from this brain. Okay, this is a motor signal. Okay, all these things we all already studied in this last previous lecture. If you want to see that, click on the link that is given in the description box. Okay, now here this continuation of this neuromuscular junctions disorder, they are basically the neuromuscular junctions disorders are classified into as first is that is autoimmune disorder. Autoimmune disorder. Autoimmune disorders. In autoimmune disorders, first there are basically two types of neuromuscular junctions disorders. That is a very famous disease. That one is myasthenia gravis. Myasthenia gravis, and another is second Labert Eaton's myasthenic syndrome. Myasthenic syndrome. Okay, here these are the two autoimmune disorder that is due to the neuromuscular junctions. Okay, here the main thing, these both are the autoimmune disorder. The main clinical difference in these two is there is defect or the impairment is present in the postsynaptic nerve. Okay, that is where is a postsynaptic, all these things you will see in this last lecture. Okay, there is postsynaptic nerve. If you want, you can see here only, that is, it means there is a nerve, it is a nerve fiber, it is a end of the nerve fiber, that is presynaptic nerve, and it is this is a muscle, it is a muscle, it is a postsynaptic, it is a presynaptic, and the space between these two, that is synaptic cleft. Okay, the presynaptic means in the myasthenia gravis the impairment is present in this postsynaptic terminal, that the defect is present in this synaptic postsynaptic. Now it is in the Leber's-Eaton myasthenia gravis, the presynaptic impairment are present. Okay, this is the main difference between this myasthenia gravis and this Leber-Eaton myasthenic syndrome. And the common thing is both they are autoimmune disorders, and both produce muscle weakness with painless, that is painless muscle weakness. Okay, these are the similar in this myasthenia gravis and this Leber-Eaton myasthenic syndrome. Now coming to the next topic, next, that is uh, congenital, that is very rare, congenital neuromuscular junctions disorder. It is very rare. Okay, it is very rare. Next, that is here it is congenital myasthenia gravis. Congenital myasthenia gravis. And last is that is due to toxins. First is there is a bacteria that is called clostridium botulinum. Okay, clostridium botulinum, it one of the bacteria that cause the neuromuscular junctions disorder. Here, next, second is that is the tubercularity. Tubercularity. This is also one of the bacteria that also cause the neuromuscular junctions disorder. Next, that is some medications are there. Medications like class of aminoglycosides. Aminoglycosides. In that also, that's too, that is gentamicin. Gentamicin. It is one of the main drug that will lead to the neuromuscular junctions disorder. And now, how this myasthenia gravis occurs, the exactly where the problem is there, and where the Leber's and Eaton myasthenic syndrome, where the exactly problem is there, and this congenital is very rare, and this toxins, this clostridium where it is act, and this tuberculari where it is act, and this gentamicin where those act in this neuromuscular junction. This is very important thing here. Okay, we have to study those things also. If you are going, if a patient is come with the symptoms, the all of this, there are some symptoms that is almost same or similar, that is painless muscle weakness is there. We can't differentiate that clinically. Okay, now we can see that, first we can see a normal neuromuscular junction. Okay, here let's suppose here this is your presynaptic nerve, this is your presynaptic nerve, it is your postsynaptic membrane of the muscle, that is muscle sarcolemma. And here whenever there is a, first we will go in detail. Okay, see this here, if it is here, these are the sodium pump, these are the sodium channel. When there is a depolarization, it means, let's see, suppose here is a vertebral column. Okay, here is our vertebral column. These are the motor neuron. This motor neural depolarization, it means the signal that comes to this motor neuron means, first the nerve that goes to depolarization. It means whenever the depolarization is there, the sodium influx in this neuron is there. Okay, it occurs. Now, when there is a sodium influx, the acetylcholine that is comes in this vesicles, and from where the acetylcholine is produced, all these things we are already studied in the last lecture. Now this acetylcholine vesicle is formed, and the acetylcholine vesicles has some special type of protein. This protein is known as synapto synaptobrevin. This protein is known as synaptobrevin, and here in the membrane of this presynaptic nerve, here is a receptor, again a protein that act as a receptor, and this protein is known as syntaxins. This protein is known as syntaxins. Now this protein, now before this is closed. Okay, this is closed with this some domain structure. Now whenever there is a sodium influx, wave of depolarization, after some time there is a opening of calcium or voltage gated calcium channel, then there is a influx of calcium in this presynaptic nerve. Now this cell has, this calcium has a capacity to open this domain like this. Okay, open this domain like this. Now this protein bind with this protein, and this protein pull the vesicle, and the acetylcholine is released from these vesicles, okay, into the synaptic cleft, and from this synaptic cleft the acetylcholine is released, and now the acetylcholine is come in this synaptic cleft. Now here on this sarcolemma of this muscle, there are some special receptor. This receptor is known as nicotinic receptor or acetylcholine receptor. Okay, acetylcholine receptor. These receptor present on this surface of this sarcolemma, that is muscle cell. Now this acetylcholine come and bind this acetylcholine receptor. This acetylcholine receptor. Now when there is a acetylcholine receptor bind with this acetylcholine, now there is a permeability open to transfer this sodium or cations into the muscle. Okay, now there is a cation influx in the muscle, and there is a calcium also influx, and there is all these things, muscle actin, myosin, There is no need to explain all this thing here. Okay, now this is a neuromuscular junction. Now what really happens here in autoimmune disorder, the myasthenia gravis, and thus 85%, the 85% of cases, there is a antibodies that form against this receptor. This is antibody that formed against this receptor. Okay, that is anti, what it is, anti acetylcholine receptor. Okay, anti acetylcholine receptor means the acetylcholine won't bind this receptor here. Due to this antibody, that is self antibody, autoimmunity fight against these receptor cells. Okay, now this is one of the cause, that is 85% of this myasthenia gravis has this type of cause, and remaining 15% of this myasthenia gravis, here is some special protein. This protein is known as that is musk protein. Here is a protein that present on the sarcolemma of this muscles, and this protein regulates the number of this acetylcholine binding receptor on the layer of this postsynaptic membrane. And this protein, if there is an antibody fight came against this protein, it means it won't regulate, or it won't function properly. The number of these acetylcholine receptor is impaired. Then also there is a muscle weakness occurs. Okay, this is myasthenia gravis. Next, coming to the Lambert-Eaton's myasthenic syndrome. Here, this Lambert-Eaton myasthenic syndrome, the calcium are blocked here. The calcium channels are do not open. Okay, due to this syntaxins proteins do not open, and this acetylcholine won't come in the synaptic cleft. Okay, this is come in this Lambert-Eaton myasthenic syndrome. Next, coming to the toxins, that is tuberculari. This tuberculari, that is a T shape that binds over here on this acetylcholine receptor. This receptor block due to this tuberculari bacteria. Okay, if any doubt is there, let me know in comment section. Please like, share and subscribe. Follow for more.